Yeah, so with that, I'll introduce Dr. Keeley MacNeill, stream ecology faculty here at UNL for the last 2 years.
No, I started in January.
Okay, so not even a year. So yeah, and Keeley got her BS at St. Olaf College and her PhD at Cornell University. And yeah, I think she's going to talk about stream ecology, and I'll let her explain the details.
Yeah, awesome. Thanks. Can everyone hear me okay? Okay, lovely. Well, thank you for being here. Hope you're all having a lovely afternoon. Today, my talk is mostly divided into 2 parts. So first, I'll talk to you a little bit about using stoichiometry as a driver of elemental cycling in high mountain streams. And the second part will be on some of my postdoc work on stream metabolic dynamics in Yellowstone National Park. So biogeochemistry is complicated, right? Our students complain about it a lot. And that's because many factors drive biogeochemical cycles. From temperature to concentrations of elements to relative concentrations of elements, precipitation, human activities. Today, I'm going to tell you about some of my personal favorite drivers and how I've thought about them in my career so far. So ecosystem ecologists have long acknowledged that examining the cycling of single elements in isolation is problematic because, is there a problem? I don't know. Can you hear me?
Yeah.
Just okay. Oh, that's maybe better. I'll try and project. Okay, so ecosystem ecologists have long acknowledged that examining the cycling of single elements in isolation is problematic because of the strong coupling of biogeochemical cycles. So multiple frameworks from metabolic theory of ecology to ecological stoichiometry all recognize the importance of considering multiple elements. Ecological stoichiometry, which I've used a lot, especially through my dissertation, is defined as how the relative availability of and organismal demand for elements drives elemental flux, storage, and metabolism in ecosystems, and is thus a really important framework for examining paired biogeochemical processes. So this graphic from NOAA gives an overview of the nitrogen cycle, which is a common way that biogeochemists think about cycling because it's a useful way of quantifying the amount of given element tied up in a specific pool or being transformed by a particular biogeochemical process. But what's hard to see from this is how many other elements are involved. So for example, nitrogen fixation, one of the main natural ways, one of the main natural inputs of N into the environment can be limited by the trace element molybdenum. While ecologists often consider carbon to nitrogen to phosphorus, there are also really interesting things happening with trace elements. So for example, the toxic effects of mercury can be mitigated by selenium, super selenium here. And the ratio of mercury to selenium controls how much mercury is retained in the food web and how much is transferred through trophic levels in stream food webs, as well as how much mercury is transported downstream. So nutrients can enter a stream from upstream, from groundwater runoff, or from the atmosphere, and they're transformed into organic form via uptake and assimilation. And from there, they make their way up through the food web to eventually be remineralized to inorganic form through excretion and decomposition. You can see here that I spent a lot of money on a graphic designer. I work in streams, which are a pretty cool system for studying biogeochemical cycling because we can measure the retention and downstream transport of elements pretty easily, and that enables us to test the importance of different drivers of biogeochemical cycling. So nutrient uptake metrics measure how nutrients move through systems in a balance of biotic and abiotic forms. And they're useful tools for examining ecosystem function and for comparing nutrient demand and processing across different streams and systems. So here we have a stream where you can see a red line meandering through, and that red line represents the path a given nutrient may take on its way through the stream. So at some point, you'll have to kind of just use your imagination a little bit here. It's not actually flying through the air. All of this is dissolved in the water column. So at some point, it's stopped by the biofilm and is being transmitted through the food web, and at other points, it's dissolved in the stream and making its way downstream. The length a given molecule travels before being removed from the water column is called the uptake length and is a common metric for measuring nutrient uptake in streams. What I wish you could see in this photo is, for one thing, the giant volcano that is back here. And for another thing, the little cabin that's off screen, that would be off screen here, which our the patron saint of this department, Alexander von Humboldt, did some of his research out of back in the couple hundred years ago. Here on the x-axis, we have stream metabolism, and on the y-axis, we have nutrient uptake velocity. On the top, we have ammonium, and on the bottom, we have phosphorus. And uptake velocity you can think of as the speed at which a given nutrient is moving towards kind of the base of the stream to be taken up by the biofilm. And as you can see, the speed of nitrogen processing here is positively correlated with metabolism. However, that's typically not the case for phosphorus, which makes sense given that nitrogen processing is mostly driven by biology, whereas phosphorus cycling is also affected by abiotic factors like sorption. So this is results based on a project that I worked on as an undergrad at St. Olaf College. Here we have nutrient enrichment, meaning the amount of nutrients we added to the stream during an experiment on the x-axis, and on the y-axis, we have uptake velocity. The plotted points or plotted lines are ammonium uptake lengths, with the solid line representing an addition where we added ammonium only, and the dotted line an addition where we added ammonium and phosphorus together. As you can see, nitrogen uptake length isn't affected whether you add nitrogen with or without phosphorus in this nitrogen limited system. However, these phosphorus uptake lengths show a different dynamic. Similar to the last graph, the solid lines represent phosphate only, and dotted lines represent phosphate and ammonium added together. And as you can see, adding nitrogen increased phosphorus uptake because it freed the biofilms responsible for taking up those nutrients from nitrogen limitation, thereby enabling them to use phosphorus. So the cellular elemental composition of organisms responsible for nutrient processing also comes into play. The different plots here represent different systems. And on the x-axis, we have the nitrogen to phosphorus supply ratio, in other words, what's dissolved in the water. And on the y-axis, we have the nitrogen to phosphorus ratio of algae. The left two graphs are some of the original studies that were done on algal versus supply nitrogen to phosphorus. And here, the organismal stoichiometry reflects the supply stoichiometry really well. It matches this one-to-one line quite well. However, as with many ecological studies, lots of research since then has found a less clean relationship between algal versus supply stoichiometry, indicating there are other factors at play. So I've just given a few examples of a few different factors that can affect stream nutrient cycling. And as you might imagine, there are plenty of others. And that's largely what my dissertation was based around. I'm only going to talk about two of my projects today for my dissertation in the interest of being able to talk about some of my postdoc work, as well as some proposed work. So first, I examined if drivers of nutrient uptake in tropical and temperate systems are different. And second, I looked at if the nitrogen to phosphorus ratio can actually drive arsenic cycling. And I'll explain the mechanisms there as I go through. So back in the 60s, Dan Jansen proposed that reduced seasonal temperature variation selects for narrower thermal tolerances and limited dispersal along tropical elevation gradients. EvoTrack, the project I was sort of tangential to, but that Dr. Steve Thomas, who was here up until this semester, explored that concept as a driver of reproductive isolation and speciation rates, as well as how varying thermal regimes drives thermal breadth and tolerance in invertebrates. But as you might imagine from everything I've just told you, I was really interested in what was happening at the base of the food web in these primary producers and heterotrophs, and what these temperature dynamics and climate variability means for nutrient processing in streams. As this middle graph from Gallenbohr et al. 2006 shows, temperature in the tropics is pretty even throughout the year, while temperate systems experience wide fluctuations in temperature. As this figure also demonstrates, elevation is important in controlling temperature, with higher elevations experiencing relatively colder temperatures. An elevation gradient also provides natural variation in other drivers important to nutrient cycling, including things like canopy cover, biomass, chlorophyll, geology, communities, and nutrient concentrations. So these are some photos of study sites in Colorado here on the left, and in Ecuador here on the right, where we leverage the spatial variation in these characteristics along an elevation gradient. So my first hypothesis for this project was that temperature is a bigger driver of nutrient uptake in tropical streams because thermal stability in tropical streams results in biofilms that are optimized to ambient temperature. And because temperate biofilms are adapted to broader thermal range because they rarely experience their optimal temperature, they're better able to maintain consistent uptake regardless of temperature. And the second hypothesis associated with this project was that variation in biofilm carbon to nitrogen to phosphorus ratios predicts variation in nutrient uptake because it reflects nutrient requirements. So you can see on the map here, we were working in the Front Range of Colorado and in the Ecuadorian Andes. And this chart just shows that they have these different sites have different ranges in environmental conditions across the elevation gradients between tropical and temperate streams. Similarly, they have similar ranges in nutrient concentrations, and generally they're all quite low in nutrients, particularly in comparison with Nebraska streams. So to be able to test if temperature is important for nutrient uptake, we need to be able to measure nutrient spiraling. And fortunately, this is much easier to do in streams than in lentic systems like wetlands and lakes. So first we add salt, which we use as a conservative tracer because it's not used by the organisms in the stream, or at least we make that assumption. And then whatever nutrients we're interested in this case, I was adding nitrogen and phosphorus both, dissolve them in a bucket and dump them in upstream. And then you measure them throughout time as they flow through. And you can do you can do a mass balance of this, but I was using the task method, which is tracer and spiraling curve characterization method by Cobino et al., which is just a kind of a modification of that. I'd be happy to talk about it more later. And basically any loss of nutrient from the water column that isn't accounted for by the conservative tracer, we attribute to biological uptake. At these sites, we also measured biofilms. So biofilms are these assemblages of algae, bacteria, and fungi growing on the bottoms of streams. And they're the organisms that are, again, responsible for uptake and making these inorganic nutrients into a form that's available to higher organisms. And with those, we measured chlorophyll A, biomass, as well as percent carbon, nitrogen, and phosphorus. Right, so one of the ways that we could look at this is just by making biplots of temperature and nutrient uptake. But because I've just told you that there are all these other things that vary along an elevation gradient, we needed a slightly more sophisticated way of looking at this. So I used structural equation modeling to tease apart the influence of these factors. So it's a technique that allows you to test a priori hypotheses based in theory about causes and effects in complex systems. So for example, you might have this sort of path diagram where you have background phosphorus and you tell the model that, okay, that affects phosphorus uptake. And we have temperature and I tell the model that, okay, I think that that's going to affect uptake through biofilm chlorophyll A accumulation. And then we have percent canopy cover. And I think that that's going to affect biofilm nitrogen to phosphorus because of the influence of shading on chlorophyll concentration and that chlorophyll molecule being really rich in nitrogen. And so we can give it this model and the data associated with it. And it can tell us the strength of these relationships as well as the direction. And just one thing to note here, I'm not going to spend a lot of time in the weeds about this, but a p-value, in this case, we often think of like 0.05, less than 0.05 is significant, and it's actually the opposite. So the higher the p-value in this case, the better the model fits, and less than 0.05 actually represents a significant misfit. So here you can see this is actually the best fit model for tropical phosphorus uptake. And so we had a pretty high p-value here, meaning it fit pretty well. We also get an R squared for each of the dependent variables and how much of the variability was explained by the model, and then the strength of the relationship and again, the directionality, whether it's positive or negative. I repeated this for all of the different nutrients, as well as, of course, for tropical and temperate streams, and have represented this as a table for lack of spending or not wanting to spend the rest of the day looking through path diagrams. So here we have temperature and chlorophyll, and we have the temperate model results and the tropical model results, as well as the different nutrients here. And here we can see that temperature and chlorophyll were important for tropical uptake, although indirectly for the tropical systems, and were not actually important for the temperate streams. And here we show that stream stoichiometry was important for most, but it was more important for temperate streams. However, biofilm stoichiometry was not as important, except for in the case of phosphorus in tropical streams. Another way that I kind of summarized this data was by looking at uptake efficiency, so we can compare how good these streams are at taking up nitrogen relative to phosphorus and plot that ratio. And you can see that tropical streams are more efficient at taking up nitrogen relative to phosphorus in comparison with these temperate streams, whereas biofilm nitrogen to phosphorus was actually lower in tropical streams. This could be due to a variety of factors, including underlying geology, higher tropical insolation enabling more efficient photosynthesis and lowering the need for nitrogen-rich photosynthesis proteins, or it could be heterotrophs playing a bigger role in the tropics. And because their growth rate increases exponentially with temperature, they may need more phosphorus-rich ribosomes overall to maintain that growth in the tropics, thus lowering their NDP. I'm going to talk a little bit about growth rate a little bit later on too. Okay, so some brief conclusions from this project. Temperature is in fact more important in driving nutrient uptake in the tropics. The nitrogen to phosphorus uptake was more efficient in the tropics, and stoichiometry does matter in driving uptake, particularly in the tropics. So we have all of this data from this elevation gradient. These graphs are just from the Ecuador sites. The top and bottom graphs are the same, but the bottom graph just has this outlier removed. And even with the outlier removed, you can see that these sites are actually pretty high in phosphorus. Through some conversations with colleagues, someone brought to our attention that we were perhaps measuring arsenic instead of phosphorus, because the method that we used to analyze phosphorus in the lab, the molybdate blue method, does not distinguish between arsenate and phosphate. So I had a moment of panic about halfway through my PhD, wondering if I had to throw out all of my data. This is important because similar to how our analytical method didn't distinguish between arsenic and phosphorus, our own cells, and in fact, most cells, can't distinguish between the two because of this shared stereochemistry. That means that the phosphate transport system can't tell the difference, and the phosphate transport system is highly conserved across kingdoms. For humans, that means a variety of factors from lung cancer to heart attacks. So to illustrate that, here's a figure of a normal phosphate molecule combining with ADP to form ATP, right, the currency of energy. However, arsenate can trick our cells into being taken up into the cell in place of phosphate. And when that happens, it can decouple both oxidative and photophosphorylation, which decreases the ability of cells to produce ATP and carry out normal metabolism. Arsenic can get into stream systems from a variety of sources, typically from any sort of significant chemical or physical weathering. So for example, volcanoes or geothermal inputs. And it's really prevalent. It affects about 200 million people's food and water supply globally. And arsenate, this form depicted here, is the form of arsenic that is typically found in oxygenated freshwaters. So these graphs show the growth curves of algae grown under different arsenic and phosphorus concentrations. So on the left are very low phosphorus, and the right are very high phosphorus. And on the right, you can see that when phosphorus isn't limiting, arsenic has no effect on arsenic, or on algal growth, excuse me. However, when phosphorus is limiting, as shown on the left graph, the relative concentration of arsenic is really important for algal growth, with the lowest growth seen in the high arsenic treatment, and then low arsenic, and then no arsenic. So we've shown the strong connection between arsenic and phosphorus, as well as the intimate linkage between nitrogen and phosphorus. So it makes sense that the three elements are all linked, right? And so I did a series of experiments in Ecuador measuring the effect of nitrogen and phosphorus on arsenic uptake by biofilms in streams. So I first wanted to conduct a broad survey of arsenic concentrations, properly distinguishing arsenic from phosphorus using what's called ICP, or inductively coupled plasma analysis, in my focal streams where I previously conducted a series of nutrient uptake experiments to try and figure out if I would in fact have to throw all of this data away. But right, that survey would only take 3 or 4 days. It takes a long time to get to Ecuador and a lot of planning. And because of the connections between arsenic and phosphorus, and by extension nitrogen, merely surveying arsenic wouldn't actually tell me anything about how arsenic affects nutrient uptake in these streams. So I also built an experiment surrounding this objective, assessing how arsenic from contaminated geothermal streams interacts with nitrogen and phosphorus cycles, hypothesizing that high nitrogen to phosphorus streams would have higher microbial arsenic uptake because of greater phosphorus demand. When I say microbial, I'm just talking about things at the base of the food web, including algae. So this was a little bit of a shot in the dark, as I had to determine which streams were high in arsenic based on educated guesses and phosphorus data, but it actually worked out really well. So I had 3 paired experiments, 2 with reaches above and below a geothermal input, and 1 with parallel streams, 1 with a geothermal input and 1 without. I was lucky enough to be able to control for temperature, pH, and a few other physical variables that often vary with geothermal input in these reaches, such that they were not a factor. However, the chemical composition varied, as was necessary to do these experiments. So I described how we would do whole stream nutrient uptake, but that involves adding the element that you're interested in to the stream, and I didn't want to dump a bunch of arsenic into these streams that I wasn't even sure had arsenic. And so we used a chamber approach instead. And you can see what happens when I'm left in the field by myself trying to carry all that gear. It just kind of explodes. So, excuse me, we have these chambers, you fill them with stream water, and then you add whatever you're interested in, phosphorus, ammonium, and nitrate in this case. One of the treatments had all 3, and then of course I had controls. And then you measure nutrient concentration over time. In this case, I measured about an hour. You put a rock with a biofilm in those chambers and to measure uptake. And I also scraped rocks throughout the stream to look at biomass, chlorophyll A, and stoichiometry. So here are some results from those experiments, which I'm pretty excited about. So the colors represent the different ambient nitrogen to phosphorus ratios, with gray being the lowest, meaning phosphorus is less limiting, nitrogen is more likely to be limiting, and the orange being high nitrogen to phosphorus, so nitrogen is more abundant relative to phosphorus. And arsenic uptake is on the y-axis, and the x-axis is the experimental nutrient amendment. And as you can see, at higher nitrogen to phosphorus in the colors, where phosphorus is more limiting relative to nitrogen, arsenic uptake is greater. And what's really cool about these results is that you can see suppression of arsenic uptake in the treatments with added phosphorus, these 2 here on the right, relative to the control, as well as the stimulation of arsenic uptake in the treatments with added nitrogen from the high N to P streams. However, for this experiment, I was only able to use the ICP analysis, which without some special modifications that I didn't do, only measures total arsenic and total phosphorus, not arsenate and phosphate. So while I was really excited about these, to double check that this cool result was actually due to arsenate and phosphate sharing the same stereochemistry, and that our results weren't confounded by the other differences in the chemical signature of geothermal streams, we repeated this experiment in Spain in the lab of Helena Guasch, who was the person who originally pointed out that we might in fact be measuring arsenic instead of phosphorus. And this work was supported by an NSF IGRIT. So for these experiments, I was able to use a modification of the molybdate blue method that actually shows you the reduced and oxidized forms of arsenic. And so you can not only distinguish phosphorus from arsenate, but you can get the different forms of arsenic, which is pretty cool. And you can see that the results held true. So the high N to P treatments had higher arsenic uptake rates than the lower N to P treatments, and that arsenic uptake was suppressed when we added phosphorus. We also measured chlorophyll A, not productivity, just chlorophyll A throughout the experiment. But because we had somewhat spotty data, I actually fed all of this into a model that uses the experimental data to predict chlorophyll concentration throughout whatever days of the experiment you tell it. And so we have ammonium at a variety of concentrations, as well as treatments with and without arsenic. And you can see that the presence of arsenic, of course, has a negative effect on chlorophyll accrual, but the higher the ammonium is, the actually more negative effect it has on chlorophyll accrual. Exciting result, nitrogen to phosphorus drives biofilm arsenic uptake. Arsenic uptake was higher when nitrogen to phosphorus was higher. Phosphorus addition suppressed arsenic uptake. Chlorophyll, this metric we used to measure algae, was negatively impacted by high nitrogen to phosphorus treatments with arsenic. And also I didn't have to throw out all of my data. It turns out that we were just measuring really high phosphorus levels. So I was pretty excited about that. Understanding controls on how arsenic moves through freshwaters is really important because in humans, arsenic consumption is associated with a multitude of human health effects. Consuming arsenic-contaminated rice or other food or water can lead to skin lesions, developmental disorders, neurological symptoms, chronic lung disease, cardiovascular disease, as well as immunological and endocrine disorders. So studying arsenic cycling and the interaction with other nutrients, perhaps used as fertilizer, is really important not only for stream ecosystem functioning, but it has major implications for human health. This is the case in developing countries. You can see the global prevalence of arsenic. This is arsenate and this is arsenite. But also right here. So in the US, about 10% of US streams are above the drinking water limit. And I think this is a really important avenue of research to pursue because phosphate transporters, like I said, are highly conserved across kingdoms. So this dynamic could play a really important but unexplored role in driving arsenic uptake in crops raised for human food, particularly in developing areas that don't have quite the infrastructure to treat their water that we luckily do. And also right here in Nebraska, where especially in the western part of the state and like along the Republican River, folks at UNL have found high arsenic concentration above the drinking water limit in both ground and surface waters. So what makes this research even more applicable to the current state of the world is that the Haber-Bosch process has provided us with a seemingly unlimited supply of nitrogen, right, leading to that phosphorus limitation. Our mineable phosphorus supplies are dwindling, and also the nitrogen to phosphorus of waterways are generally increasing. So one of the projects that I would like to work on, I have a proposal written and plan to submit it at some point, would be to develop a framework assessing locations at risk of crop arsenic contamination based on overlap in high nitrogen to phosphorus and significant surface water arsenic. To do that, I would leverage public surface and groundwater nutrient databases in tandem with extensive collated literature on arsenic concentration. And also, I would like to experimentally test using a series of both field surveys and greenhouse experiments. If growth rate, again, this high growth rate means high demand for phosphorus and therefore probably also high arsenic uptake, although that hasn't been tested, and relative nitrogen to phosphorus drive crop yield and arsenic retention using, like I said, surveys and a greenhouse experiment. Okay, moving on to part two, which I did my postdoc out at Oregon State with Dana Warren. This is the collaboration between the folks listed here at Oregon State and then Colton Baxter and the PhD student Jeremy Brooks at Idaho State in Yellowstone National Park. So we conducted this research in the northern range of Yellowstone, shown here in an inset map in the very northwestern corner of Wyoming. You can see this is right along the Montana border. Many of you are probably familiar with this location, either from having visited there as a tourist or perhaps having heard about this location as where we reintroduced wolves in the mid-1990s after their absence for almost a century. And where it is thought that the reintroduction has cascading effects on the surrounding ecosystem. So one of the common stories that has come out of this was featured by National Geographic called How Wolves Save Rivers, which posits that after the reintroduction of wolves, riparian ecosystems increased in diversity and productivity, as shown here in the right-hand panel. However, there's been debate about whether or not and to what degree these consequences actually occurred, in part because, while some streams like Antelope Creek, shown here, have indeed regrown their alder and willow communities, other creeks, like Rose Creek, have not regrown at all. So there's been a broad array of interdisciplinary work assessing why this phenomenon is occurring in such a patchy way across the landscape. This includes investigations into the broader predator community, so not only including wolves, but also grizzlies and cougars, which have increased in numbers over the last 30 years also. It includes the prolonged absence of beavers, particularly in these sites. We never saw any evidence of beavers in the sites we were working in. It includes changes in the physical structure of the streams. It includes a pretty significant increase in bison population. Some have started comparing the Lamar Valley, where a lot of our work was, to just feedlots, which I don't think it's quite that bad, but they are a significant presence there, as well as the changing climate of the past several decades. So all of these things are thought to be interacting and contributing to this patchy distribution of riparian willow and alder communities throughout the northern range. And while there's been considerable research into this phenomenon, there's been relatively little research assessing functions like productivity and diversity in these ecosystems, particularly for the aquatic portion. And this is the gap that our research aims to fill. So for most of the rest of the talk, I'll be sharing some of the results of our research on the productivity of the aquatic portion of these streams. So while we originally thought of these streams as categorically high vegetation or low vegetation or willow alder versus grass sedge dominated, our findings have led us to think of them as a mosaic of these ecosystem states and potential trajectories. So we selected 8 headwater streams in the northern range between 2018 and 2021, and all were selected to be in alluvial valley segments where they have the potential to recruit and sustain willow and alder communities. So we also used historic photos to guide us in the selection. The Park Service has a really nice archive of these. And you can see in the top left here, 3 of the 8 of these streams went intermittent part of the way through the summer. They just lost flow completely. And while this is ecologically relevant and will aid in our interpretations of data down the road, today it's really hard to model those streams when there's no flow and as they're going to that point of no flow. So today I'll just be showing you results from our 5 perennial streams which had water throughout the year. To guide our investigations, we used this diagram from Colton Baxter, one of our collaborators, in a paper he published in 2005 on ecological linkages between land and water. And briefly, this shows that changes in riparian plant communities can influence the amount of terrestrial organic matter, terrestrial invertebrates entering stream ecosystems, which has consequences for fish and invertebrates and even the primary producers. Many aquatic inverts have life histories where they emerge into riparian insects, which has consequences for terrestrial insectivores like birds and bats. While our research includes estimates of all of these different compartments of the ecosystem, our work remains preliminary, and today I'll just focus on the aquatic portion of the ecosystem and particularly on productivity, where I've done most of my work. So these are the 5 study sites that did not stop flowing in the middle of the summer. So on the left side are geode, antelope, and East Fork black-tailed deer creek, and those are the creeks dominated by either willow or alder vegetation. And on the right are the open creeks with sedge, sedge, and grass-dominated vegetation, rose, and Lost Creek. So stream food webs are connected to the organisms at the base of the food web that take carbon and nutrients and again transform them into these forms that are available for higher trophic levels. The way that we're able to model metabolism or the combination of GPP or gross primary production and ER or ecosystem restoration is by monitoring changes in oxygen concentration relative to other physical factors. We only want to focus on the changes in oxygen concentration due to the heterotrophic and autotrophic activity, but we do account for those physical processes. However, we're really just using oxygen as a proxy for carbon, which is in balance with oxygen during photosynthesis and respiration and serves as the energy basis of stream food webs. So looking at the balance of GPP and ER can give us an idea of where that energy is coming from and also the role that a given stream plays in a watershed in storing versus transporting carbon. So we estimated a variety of metrics out there. We deployed mini dot loggers in these streams and left them out there all year long. I called up PME, the company that makes mini dots, and was like, hey, do you think, can I leave them out there all year? Will it be okay? And they're like, we have no idea, but we'd love it if you could tell us. And we did, and they were fine. And these loggers continuously monitor temperature. We used a different logger to monitor photosynthetically active radiation, although we ended up modeling this for metabolism. It's a little bit easier to get an estimate based on latitude and time of year than it is to use a patchy logger and try and integrate that for a given stream reach. The PME loggers also log oxygen, and then throughout the year we took, and especially the summer growing season, we took estimates of basal resource biomass, looking at not only these biofilms that I was talking about in previous research, but also the organisms and organic matter that are suspended in the water column, as well as the fine particulate organic matter that settles to the bottom of a stream. We modeled metabolism using an R package called Stream Metabolizer, which is an inverse Bayesian modeling technique. And K600 is a parameter that is really important for this, and it describes gas reaeration in the stream. So the amount of atmospheric exchange of gas you might have, which of course, when you're trying to estimate the biotic portion of that, we have to be able to account for that. You can model that particularly in streams that aren't very turbid. And so we attempted that to start with. And in the cases where that didn't work, I had some preliminary data from the first couple of months to play with. In the cases where that didn't work, we went back to the field and empirically estimated it using gas tracer additions, which are similar to the nutrient additions, but using gases. So several previous studies have examined the effects of canopy cover on stream metabolism, citing light in open canopies and organic matter inputs in closed canopies as the most common mechanistic drivers of metabolic dynamics. In this study shown here, called LINKS, of 17 streams across a variety of landscapes and latitudes, they found fairly strong evidence that GPP is higher in open canopied streams due to light, while ER is higher in closed canopy streams due to increased organic matter. And not surprising, based on the first two panels, is that the ratio of GPP to ER is much higher in open canopied streams. So as one of the most initially obvious variables in the streams we were working in Yellowstone is canopy cover. We were wondering about similar mechanistic drivers of metabolism. And on the right here will be our data. So on the left is the LINKS plot for the next several plots. And you can see that the sites in the northern range fit really nicely with the LINKS data in terms of GPP, showing a higher GPP in open canopied streams. In the case of ecosystem respiration, our data didn't follow the same pattern as the 17 sites in the LINKS experiment, not really showing differences in ecosystem respiration between high veg and low veg streams. This could in part be because bison are, for example, supplementing organic matter inputs in the open streams, as well as Lost in particular being downstream from a pretty major wetland area. I think this is, may not look very interesting, but I actually think it is a really interesting result because these sites, which are so geographically close, can break this broad scale pattern of links, showing that regional differences can be more important than traditional drivers. And another important consideration here is that the two open sites both remained open year round, whereas the three closed sites iced over and two of the three of them froze solid. And so we're not flowing at all. And so there's no signal from Antelope or Geode Creek in the middle of the winter. Had they not frozen over, they likely would have exceeded the two open sites in ER. In spite of the fact that ER ecosystem respiration in the Yellowstone streams did not follow the same pattern as the LINKS data, the ratio of GPP to ER did. However, the summary of the data overlooks seasonal dynamics and also canopy cover is not all the same. So I think that in this case of the LINKS data, all of the closed canopy sites were forested streams and ours were not. So given the background info on the streams as a mosaic rather than categorically high versus low vegetation, and given that our data did not follow the same patterns as previous studies, we wondered if we could use this mosaic approach to better understand the metabolic dynamics in these streams within a really relatively small geographic region by also considering some of the other potential drivers like discharge and its variability, connectivity with the floodplain, as well as other upstream influences. And we can do that using this figure from Bernhardt et al. 2018, a paper that explores the various drivers of GPP and ER. So here GPP is on the x-axis and ER is on the y-axis. Data along this one-to-one line indicates that GPP and ER are similar as it is and data below it indicates that ER is greater than GPP as it is in most streams. Note that the lower the ER is, the higher it actually is because we're looking at oxygen consumption. And so you can see these 5 drivers depicted here. You can see that light and scouring flows are kind of the two main ones, with scouring flows knocking back both ecosystem respiration and GPP because they prevent these organisms at the base of the food web from kind of establishing, holding on to anything and doing their job. Light increases GPP as well as autotrophic respiration. Heat and nutrients have somewhat similar effects, although affect ecosystem respiration slightly more strongly than GPP. And then DOC and sediment increase ecosystem respiration by supplementing carbon, but decrease GPP by minimizing light. So starting to get into our results, in the bottom left here is Lost Creek from this study with the different colored points representing different seasons. So yellow is summer 2019, maroon is fall, gray is winter, green is spring, and blue is the following summer 2020. Here you can see all 5 of our study sites with the willow alder dominated sites on the top row and the sedge grass dominated sites on the bottom row. And you can see these sedge grass sites have more days approaching net autotrophy, right above this 1-to-1 line, or at least right on the 1-to-1 line. But in general, there aren't striking consistent categorical differences in metabolic dynamics between our limited replicates of willow alder versus sedge and grass dominated sites. So we can turn to this Bernhardt et al. Figure on the right to consider each of these drivers across the sites. So for the rest of the talk, the figure on the left will remain the same. We'll just dissect it a little bit. So first we can look at light, which again increases GPP in autotrophic respiration. These measurements of photosynthetically active radiation on the y-axis were taken as an average throughout the stream channel during the summer. And in general, surprise, the open sites have more light. And this is actually reflected in the metabolic data in the extension of the data into the downward and right portions of the plots in Lost and Rose, shown here by the blue arrows. If we look at Blacktail Deer and Lost Creek, these 2 sites, especially in spring, we can see high peaks of both GPP and ER. Both of these streams have a really high connectivity with the floodplain, which provides a strong signal from both autotrophs and heterotrophs associated with additional surface area, substrate surface area provided by the grasses. In Lost and Rose, which remained open all winter, GPP ranges from about 1 to 3 grams oxygen per meter squared per day in the winter, represented by the gray points, whereas GPP is basically 0 in the winter in the closed sites on the top, which froze and obviously had no light. So what's interesting here is that while these sites have the same capacity, they've diverged. So it's obvious that these open sites would have greater GPP, but in the spring, a closed site that becomes more connected to the floodplain gains access to extra substrate as well as extra light and actually shows greater GPP than a more open site. So Rose Creek here had the highest flow and also the most mobile substrate. It had smaller gravelly bits as opposed to largely a sandy clay bottom on the rest of the streams. And you can see that this appears to knock back GPP and ER, especially in the spring when flows are the highest. Wetlands also mitigate flows. So Blacktail Deer, Geode, and Lost Creek all have associated wetlands, and those wetlands appear to help mediate the scouring and reduce that energy flowing through the stream, therefore supporting higher productivity. Lost and Rose are 2 open sites. On the bottom are similar on the GPP axis, this x-axis, but Lost is much higher in ecosystem respiration, likely because Rose is not storing this organic matter, so it's not available for respiration because of constant scouring. And lastly, before I talk about just a couple of proposed projects, we have DOC and sediment, which decrease GPP and increase ecosystem respiration. So we don't have good temporal resolution for either sediment or DOC, but I'll share with you what I have. So Geode and Lost Creek are both downstream from historic beaver wetlands. And the range in GPP is much lower in Geode, but most summer data didn't actually converge. It didn't work in the model, so I had to throw it out, and so it's not displayed here. One potentially rich source of organic carbon is from benthic organic matter, which is, again, that like fine organic material that kind of settles to the bottom of the stream, and it's represented here in the top graph. And you can see that Rose has the lowest of all of the sites, which is reflected in the metabolism data with the downward shifts or shifts towards ER in all except Rose. And this is further corroborated by our dissolved organic carbon data, where again, Rose was the lowest. And you can see that at least in Blacktail Deer, Geode, and Lost, this dynamic seems to be particularly important in the spring in the green data, with the spring flood flushing accumulated organic matter in from the floodplain. So just a quick summary, we saw no consistent categorical differences in stream metabolism between high and low vegetation sites. We really need to consider the full picture here. So not only seasonal differences in light, I talked about Blacktail Deer, a high vegetation site that has more comparable light to a low vegetation site in the fall and spring, but also flow, nutrients, carbon sources, and temperatures. And these dynamics in GPP and ER are driven by local features and actually the historic stream trajectory. Also, metabolic dynamics across the northern range of Yellowstone are as variable as studies comparing streams across the world. And these fingerprints are those data by plots show important variation within categories. So this presentation hopefully doesn't come off as just an ad for this Bernhardt et al. 2018 and her conceptual model that works really well. That graphic was meant to interpret broad scale patterns across different locations, but these sites occupy really different parts of the figure, even though they're in a really small geographic region, either because of this kind of mosaic concept that I've talked about or because of seasonality. So many of you are probably familiar with the June 2022 floods this last summer that devastated human infrastructure in and around the park and also affected and really kind of washed out lots of our streams. This fall, we applied for and received a rapid grant from NSF to support future work looking at the effect of these terrestrial dynamics and the effect of this patchy mosaic on stream ecosystem resilience to disturbance. So you can see here, one of our streams was like this perfect meandering stream, and it just got totally washed out here. So we're going to go back and measure these channel morphology characteristics. We've already redeployed loggers there, and so we'll be re-examining these dynamics. I already mentioned the future work I'd like to do with arsenic, but another project that I'm working on here in Nebraska is looking at antibiotic presence in streams across Nebraska with particular respect to different land use types. Looking kind of a continuation of some work done by Shannon Bartelt Hunt and others here at the University of Nebraska, looking at both antibiotics from CAFOs and animals and whatnot, and also human antibiotic use. And we'll be comparing antibiotic concentration above and below sources, including manure applied agriculture and concentrated animal feeding operations, as well as urban streams. And I've also had a lot of really interesting conversations with colleagues here at UNL about potential collaborations that I really look forward to further developing. With that, I'd be happy to take any questions.
Arsenic. So my question is, did you look at basal rates of arsenic uptake in the biofilms between streams that had arsenic in them and streams that didn't?
I did. As in adding arsenic to streams that I did not. No, that would have been really cool. But again, that gets at like having to add arsenic, and all of this was done in the field in the
in a bucket experiment.
Yeah, in the lab. No, I guess I would have done that in the Spain experiment. No, we didn't do that. That would have been really cool.
You've read a lot about this. Do you think it would differ or no?
I would imagine that maybe, even though this is so conserved across taxa, I would imagine that maybe the taxa that are arsenic adapted have gotten a little bit better at dealing with it and maybe recognizing it. So maybe a naive system would be a little bit better at taking it up and therefore be more negatively impacted by it. Yeah, that'd be really cool to look at.
Well, thanks.
I'll follow up on the arsenic because I don't think about arsenic much.
No
So from an environmental health point of view, would you like the stream to be increased uptake of arsenic because then it's getting in the food chain?
Yeah.
Or do you want not to have the uptake and then it would stay in the water column and just go downstream? What's the strategy, or is it more complicated?
Yeah, some people are, looking at this potentially at not the nitrogen to phosphorus dynamics. To my knowledge, no one else has looked at that, but in terms of relative arsenic uptake rates, I think it depends largely on where you are, right? What's downstream of you. It certainly can be used as a mitigation tactic, and even if it's not biotic uptake, they can use like you've maybe heard of these floating treatment wetlands that use lanthanum candles to kind of bind phosphorus, and you can use similar strategies to bind bind arsenic and get it out of the system, but then you have to deal with that. Right, in terms of the algae, I don't think that it necessarily impacts them terribly in terms of broad scale food web effects. I have some research on the whole food web that I wanted to share, but just didn't have time today. But it doesn't transfer well. It doesn't continue to transfer well through trophic levels. So it bioaccumulates, but doesn't biomagnify. So I think it really just depends on where you are in terms of whether that's an effective way to deal with arsenic contamination.
Thanks, Keeley. That was really cool. Do you know of any other elements that have a similar structure to nitrogen or phosphorus to kind of do this toxin trickery that arsenic does with phosphorus?
Not that I know of, no.
Kind of a way downstream issue. That 10% statistic, is that found in streams or across the nation in terms of waterway arsenic population? And do you, if it is across the nation, do you also think that's because of a lot of this country, especially where I'm from, the Northeast, does not have separate sewer and stormwater runoff systems? So basically you could have the Hudson River be perfectly clean, swimmable one day, and then the next week be completely polluted because of rain in terms of arsenic and other heavy metals.
So let me double check that I'm understanding you. So the 10% statistic you're talking about.
Yeah, and that map that was presented on the arsenic.
Globally, yeah.
Yes.
So the 10% statistic is just, it's from the US Geological Survey. So that's just specific to the US. It can be super patchy throughout the US. I should have shown a map of just the US alone, but right, there are little pockets, although of course not everywhere has been estimated. And certainly, right, like mine waste is a particular problem. And if there's a big flood, that can have tremendous consequences for downstream ecosystems when all of that suddenly gets flushed in. Does that answer your question?
In particular, I'm wondering how much contribution is the in a lot of our cities, especially Lincoln, actually, not having separate wastewater systems in arsenic, if you're familiar with that issue, or other heavy metals in the water supply.
I mean, certainly, right, in older cities with like leaky infrastructure, it could result in arsenic leaching into our water supply. And or if there's some sort of arsenic source within a city, likewise that could leach out into. But arsenic sources are typically either from, like I said, geothermal or volcanic activity, or from upwelling deep in the earth, or from things like mining waste.
I have one too when you're done, Dave.
Okay. Why don't you go over there and then we can run back. At least some alders are end fixers and your willows are not. So does that enter in these riparian systems? Is there much end fixation? Does that screw up with your terrestrial stoichiometry?
Yeah, you know, we've looked at that. I again didn't present those results today, partly because we're still interpreting them. So right, you're right, alders are really important end fixers, and I tried to measure end concentration downstream from a given tree in all of these reaches and couldn't really suss it out that way. But not only looking at end fixation from these alders, we've been looking at the riparian plant community in the grasslands, and there are lots of clovers and other species that play a really important role in end fixation. So I don't think it's necessarily a categorical difference in the streams with alders versus the like upland grassland streams, but certainly that's an important component, particularly given that these streams are all really low in nitrogen and they're all quite high in phosphorus. And so the stoichiometry story isn't really that interesting for this system because they're never phosphorus limited, they're always nitrogen limited. And then we also looked at end fixers within the stream, looking at the algal communities. And there we did see a slight difference between these different categories of streams with more end fixation, or at least more end fixers being present in the open sites.
So did wolves save rivers?
Well, they're important, but they're not the whole story.
So, yeah, and maybe it's a dumb question because I admit I didn't follow everything because it's a little out of my league, but what does your data tell us, if anything, about wolves or the restoration of large carnivores in Yellowstone?
Yeah, I mean, I wish I'd had, maybe I should have just talked only about that project, to incorporate aspects from, the higher trophic levels within the stream and then the terrestrial aquatic exchanges. I think we're still putting the story together. Certainly it's not just wolves, right? I think that there are too many other things that vary, like, not only other carnivores, but again, also climate change and the structure of the channel morphology changing, and also the bison presence. That's a huge factor in these streams, right? Some of these are just getting absolutely mowed down, and then some of the other streams are kind of where, there's a lone bull every now and then, but it's not the huge herds that we see. So they are important, and a more nuanced story is TBD.
Yeah, I would agree with that. Also, it was interesting, if I understood it, it seemed like the effects you were seeing were just highly variable across Yellowstone.
Yeah.
And there's definitely been talk about the effect of wolves on prey and vegetation being highly variable, and that they were really important in some areas and not so much